Lecture series, for your information, is the fourth Tuesday every academic year in October, November, February, March, this month, and April. April's talk is going to be me, so I'm ending the STEM lecture series for this year, and my talk on April 29th will be about Lewis Carroll. But I'm a mathematician, so what the heck does Lewis Carroll, who wrote Alice in Wonderland and Through the Looking Glass, have to do with mathematics, and what hidden secrets are in his writings? Well, come to my talk next month. It'll be heavily advertised as a screensaver on Delegate, and by email, and by posters plastered all around campus. So I would like to circulate a roster so I know who attended this talk. I'll start it right here. It should circulate this side of the auditorium, then transfer to the other side, and eventually make it up to Eric, and I'll get it back from Eric. So I want everyone to have a chance to sign the roster. There's a place for your name, the professor for whom you might get extra credit for attending this talk, if appropriate, or if not, leave it blank, and the class. If you're not getting extra credit, we merely want your name so we have an idea of who attended. So I'll start it with Mike, and it should circulate around the auditorium. If you signed it already, just pass it to the next person. It should migrate to the back and then circulate this other side and eventually make it to Eric. A synopsis of the talk: neurological diseases, like Parkinson's disease, for example, Michael J. Fox, account for more years lost to disability and mortality than any other disease. Therefore, understanding the causes of these disorders and finding novel treatments are of great importance. The advent of genetic manipulation in cells and laboratory organisms has provided neuroscientists with powerful tools for modeling, studying, and treating such diseases, so customized treatments. As a matter of fact, they discussed that in a recent article in the Philadelphia Inquirer. So this lecture will describe recent research into several disorders of neurodevelopment and illustrate how these studies are leading to cutting-edge new therapies. Our speaker, Dr. Nicole Sunin, is currently a third-year fellow in the Penn Port program. She received her PhD in neuroscience from Baylor College of Medicine after completing her Bachelor of Science in molecular biology and genetics in Canada. Her research at Children's Hospital of Philadelphia, CHOP, centers on molecular mechanisms of aberrant neurodevelopment as she attempts to uncover novel therapeutic targets. So without further introduction, welcome Dr. Nicole Sunin.
Thank you, Dr. Pulpus. Can everybody hear me now? Is this even on?
Yes.
Oh, good, because I don't hear an echo, which is fantastic. It's really nice. Yeah, so I'm told this is a really good turnout, so I'm feeling rather popular, and I hope I live up to your expectations. As Dr. Pulpus said, I am a Penn Port Fellow at the University of Pennsylvania. What that means is that I do postdoctoral research. I got my PhD already, but I'm doing research in someone else's lab. It's another kind of training program before you move into, say, a professorship level, which would be the traditional career path. And in the meantime, I'm also teaching. So if you've seen me around the halls here before, it's because I've actually taught a couple sections of anatomy and physiology here at Delaware County Community College. So this is a really neat program where I get to both teach and do research. And so that's one of my passions. I get to combine my two favorite things. So this talk is about neurological disease and also a little bit about my career path and how, if you like what you see, if it's something you think you might want to do, you can, I hope I can give you some guidance on how you might want to get there. But let's start at the very beginning and talk about what a neurological disease is. So it's any disruption of the nervous system in its structure, its function, or both. And currently there's already more than 600 recognized neurological diseases. So some of the ones you might have heard of before are things like Alzheimer's disease, Parkinson's was mentioned, epilepsy, multiple sclerosis. How many of you have heard of at least one of these? Yeah, everybody has heard of at least one of these. It's kind of a hot topic these days. It's where a lot of research is going on. And it's really important to study these things. But where I started out, where I became interested in it, is actually many steps removed. I didn't start out being incredibly interested in neuroscience. I didn't come out being incredibly interested even in biology in the beginning. But my interest started around the time I started high school with this guy. I grew up with dogs, golden retrievers specifically. Love them. If you have a pet at home, you know exactly what I mean. They're loyal, they're sweet, they're fun, they're hilarious, they do some weird things. This is Sunny. He used to snarl on one side if you scratched his face on that side. We loved him terribly. So we got him as a puppy when just about the time that I was going into high school. And, he was a great puppy. But he didn't play and run like normal puppies do. He seemed to be in pain when he did that. And we took him to the vet and we tried to see what was wrong with him. And it's not a neurological disease he had, but he had this problem known as hip dysplasia. It's actually really common in several breeds of dog, large breeds of dog. And essentially what that means is that the leg bone, the femur, that top that fits into the hip, make it all rotate nice and move around, doesn't fit right in his case. On both sides, both his right hip and his left hip in the back. It just doesn't fit right, and so it caused him a lot of pain every time he was running and doing things. So we took him 2.5 hours away from my hometown. I grew up in a small town in Canada. And we took him 2.5 hour drive away to the University of Guelph, where the local vet college was, to have surgeries done. On one hip, they literally cut his hip in 3 places and rebuilt everything around it to fit. And in the other hip, they cut off the top in order to fit in. So we went through all of these surgeries. There was a long recovery time. It was, really traumatic. But in the end, he was actually a really happy-go-lucky dog. He lived till he was 13 years old, which is the oldest of any of our dogs that we had before. We had golden retrievers the whole time growing up. So it does work. This was really good that we could do this. But at the same time, I was starting to take courses in biology in high school, and it was really bothering me. Why was this necessary for a 6-month-old puppy to have such horrible surgeries? Like he lost his childhood, right? And that really bothered me. And one of the things I learned in high school biology was about what people would call essential dogma, and that Sunny's hip dysplasia was actually a genetic problem. It was a genetic disease. So even though his parents didn't have this problem, somewhere in this process of DNA being read to RNA, being read to protein to give your traits, like your hair color and your eye color, and whether or not your hip fits properly with your femur bone, somewhere along the line, something went wrong. There was a mistake, there was a mutation, and that's what gave us disease. And this is something that I got really interested in. I thought, well, how is this possible that at anywhere in this process you can get disease? So I went to college, and lo and behold, so this is where I grew up, that little red dot is my small town, Chatham, about 45,000 people. And this is Guelph, 2.5 hours away. So I decided to go exactly where we took him for all of his surgeries for a couple of reasons. One, I was very interested in genetics now, and the chemistry, the molecular biology behind these types of diseases. And they had a program in molecular biology and genetics. It was the only one in the country that combined the two, and that was really exciting. The other thing is I thought, well, maybe I want to be a veterinarian. Maybe I want to do these kinds of surgeries. I want to help out people's dogs. And that's where the vet college was, so that's where I went. And I took a bunch of different courses. Lots of chemistry, lots of biology, genetics, development, physiology as well. I also took some psychology courses, mostly just for fun. I thought it would be kind of cool to see how the mind works. And I really liked the psychology courses, but it didn't get at the mechanism of why things were the way they were. And that's what I was missing. I really wanted to know way down on the molecule level, on the DNA level, why things were the way they were. I also took a couple classes and some internships in laboratory research. and found that I really liked it. I really didn't think I would. I thought that people that worked in laboratories, scientists, were dirty and antisocial and didn't know how to talk to other people, and I thought it would be really isolating. But in fact, some of my best friendships have been from people I worked in the lab with. I went out and I played ultimate frisbee with the master's students in the lab that I worked with. I had classmates that I'm still friends with that would throw parties with themes like "Don't you hate pants?" Okay, so these were actually fun people, and I really liked working in the lab. I liked figuring things out for myself, doing that discovery process, asking questions, and the neat techniques, getting down to the nitty-gritty of how things work. I really enjoyed that. So I started to think, well, I like the psychology, but it's not telling me exactly what I want to know down on the level I want to know. And I really still like research, and I had some fantastic professors, and I thought I kind of like teaching at the college level too. So I'm going to go ahead and I'm going to go get my PhD, and I'm going to apply to grad school. So I applied to several graduate programs. You have to take a couple standardized tests. You have to have some good grades. You go on interviews. It can be a little bit traumatic in that final semester of your undergraduate lifetime. But I moved to Texas. I got accepted into the Baylor College of Medicine program in neuroscience. And one of the reasons, so I moved all the way up here, from here all the way down to Texas, for a couple of reasons. One of the reasons is the program was very well ranked at the time. It was really highly respected, and I thought I could get a really good education there. And the other thing is that it's part of this Texas Medical Center that's all hospitals and all biomedical research buildings. And there's a lot of collaboration. There's a lot of crossover between what's going on in the laboratory and how that can be translated into helping patients in the clinic and in the hospitals. And that was something that really appealed to me. So my laboratory work was in conjunction, my thesis work, with the Baylor College of Medicine and Texas Children's Hospital, which is where I actually conducted all of my experiments. I took a lot of courses in neuroscience while I was there as I started my research. And one of the #1 things I learned is that brains are highly, highly organized. They're not just a big block of tissue, okay? If this is a picture of a human brain here, how many of you have seen a picture of a human brain? It's all wrinkly? Yeah, okay. So it's divided up into several functional areas, and each one of those areas has a very specific structure for its specific function. So if, for example, if we were to zoom in on a little piece of this neocortex here, that's where a lot of your higher order functioning happens, and take a look at it, you'll see a bunch of different cells. Each one of these colors represents a different kind of neuron. And you'll notice that they're layered in a very specific fashion. So here's this yellow type of neuron, and it's always going to be found in this layer here. Okay, so they're connected up very specifically and highly organized in order to do what they're supposed to do. Most of you know that neurons make connections, yes? Yes, okay. So if we zoom in on some of these connections up here, zoom in even further, we get down to the nitty-gritty into how neurons communicate between each other. On the one side, you have usually an axon, and then there's a synapse, a synaptic cleft, and then you have your dendrite on the other side. And this is how neurons communicate with each other. So they send neurotransmitters across this cleft and bind to very specific receptors in order to pass on their information. They're organized into circuits, into electrical circuits. And it's worth noting that you have about 86 billion neurons in your brain, and each one of those can make up to thousands of connections. So there's a lot going on. There's never really a simple circuit structure. And it's important to know that sometimes, if neurons are constantly passing on all of their information, it gets really noisy and you can't pick out any one message. So imagine that every individual neuron in your brain is a different radio station. And you turn on all of those radios in the same room to a different station. Can you make out any one song? No. Okay. But if you turn down the volume on some of those stations, or turn up the volume on other ones, then you start to pick out a message. So the decision as to whether this information is passed on or not, turning down or turning up that volume, is a balance between excitation and inhibition. So some of these neurotransmitters, some of these receptors are going to turn down the volume. They're going to be inhibitory. And some others are going to turn up the volume. They're going to be excitatory. Does that make sense? Okay. And throughout your brain, throughout all of your connections, you have this balance between excitation and inhibition. When things are out of balance is when you get these neurological diseases. So it's really a problem of connections, either how they're built or how they are being rewired, and why things, you're either getting the wrong message through or there's chaos and you're not getting a message at all. Okay, so this is the basis of neurological disease, is the problem with neural communication. And it got me thinking as I was taking these classes and starting to do my research, that how these circuits are built in the first place is really important. Because if they're built wrong, then you're going to have this problem with communication, where you're getting the wrong message or too many messages and you can't pull them out. So I got really, really interested in neural development. So here's a list of a couple diseases you might have heard of that are specific to neural development. Who's heard of autism? Being highly studied right now. How about Down syndrome? They have a lot of other problems, children with Down syndrome. And most of these diseases you'll see in children before you see them in adults, because these are the kinds of problems that are happening during at an embryo stage while the brain is just forming, just making all of its initial connections. So this is something I became really, really interested in. All right. Ah, yes. Sorry. So the other thing I wanted to mention about this is that one of the hallmarks you'll see with a lot of these neural developmental diseases is that these children tend to be really devastated by them. They tend to have learning disabilities. They tend to have social disabilities. They might have motor coordination problems. And another hallmark that they tend to have is epilepsy. And epilepsy we usually define as an imbalance between your excitation and your inhibition. There's more excitation or not enough inhibition. And what happens in those cases is all of those radio stations get turned on at the same level and you can't make out anything. Okay? The other problem with having these seizures is that typically you'll have more than one. So one seizure tends to lead to another seizure, tends to lead to another seizure. And this is really important because a lot of people don't seem to realize how prevalent epilepsy is. So there's probably about 100 people in this room, about that. Chances are one or two of you is actually diagnosed with epilepsy already or will be later in your lifetime. So it's that prevalent. It also happens to be really most common in children under 2 and in the elderly, and this is something that really struck me as being incredibly important. Children under 2, especially since about a third of them don't respond to the medications that are available. So epilepsy drugs that are out there, there's a lot of them to try. If you've tried 2 of them and neither of them work for you, you're considered to be drug resistant, and there's not much you can do to control those seizures. 50% of the drug resistant cases in children are due to what we call malformations of brain development. So there's usually a structural developmental problem that you can see. So that's where my motivation came in. There is a real need to find new treatments, to figure out what the mechanisms are, and to find more targeted therapies for these poor people, for these poor children. I wanted to help these families with their burden. And so I'm going to talk about 2 different research projects that I've been working on. The first one is about the disease cortical dysplasia, and this is what I spent my PhD thesis doing. This is a disease that has unknown causes. Okay, and we used a certain approach. We call this an in vivo approach, which I'll talk about in more detail in just a second. And then I'm going to talk about what I'm working on now, what I'm just starting to work on, which has to do with one single patient that a colleague of mine sees in the clinic. and what I'm going to do using an in vitro approach to try to help him. Okay, so let's break down what I mean by in vivo and in vitro. Has anybody actually heard those terms before? Okay, a few of you at least. So when we talk about in vivo, we're talking about using a whole organism. This can be a person, it can be a worm, it can be a mouse. When we talk about in vitro, we're talking about isolating a piece of the organism and looking at it in a dish. And the reason you use these two different approaches are for different reasons. An in vivo approach allows you to ask big picture questions. So I could ask the question, if I give this mouse cigarettes, will it get addicted? And it doesn't matter why he gets addicted, but because everything's all intact, it's a whole mouse, I can look and I can say that mouse is addicted to cigarettes, or not, okay? On the contrary, if you look in vitro, I can ask a question like, if I take these neurons out of the mouse and grow them in a dish and give them nicotine from cigarettes, does it change their connections? So now I'm getting more down into the nitty-gritty of how things work. And the two are very complementary to each other, but you use them for different things. As I already mentioned, my thesis work, looking at cortical dysplasia, we use this in vivo approach. So this is where I started my graduate work. I had taken a bunch of classes down in Houston at Baylor College of Medicine, and it was time for me to choose a lab. And I chose a collaborative lab between Dr. Ann Anderson and Dr. Gabriella Angelo. These are two very bright women who were working together on cortical dysplasia. So cortical dysplasia, these patients, these children come in with seizures, they come in with epilepsy, they can't be treated with drugs, the drugs that are available don't work for them, and so the only thing that we can do for them is to actually cut out the piece of the brain that is causing their seizures. So what you'll see here in these pictures are pictures of normal brain. Zoomed in here, you can see all of the neurons and how they're layered in a really nice, organized fashion. And then you can see it from a patient. Do you see those layers anymore? No. See how there's like big gaps missing? Notice too that the neurons don't look the same. They're really big. And this was something that was that they were looking at, and they wanted to know what's causing this. You know, if we can prevent it, or if we can target a treatment, then we don't have to cut out pieces of their brain. Among other things, there are pieces of their brain that you can't do without, that you simply can't cut out. And so it's not a very good treatment that's available for these children. So when I joined the lab, what they were doing was this really cool technique where they would laser capture individual neurons from this tissue that they were getting from surgery. They would suck it out, and then they'd go look at gene expression. So basically what they were looking at is they looked between diseased brain and normal brain and see what genes, what proteins are there more or less of as compared to normal. And what they found in these big neurons, these abnormal neurons, was this growth pathway. We call it the mTOR pathway. You'll find it in all of your cells. What it is it's a cascade, so this molecule does something to turn on this molecule, does something to turn on this molecule, and so on and so forth, and the outcome is cellular growth. Well, neurons are kind of interesting. Usually when cells grow, they get bigger, and they divide, and they make more cells. The vast majority of neurons don't do that. So this kind of makes sense. Maybe that's why these neurons are really big. The other thing I want to point out is that there are regulators at the top of this pathway. P10 is one of these regulators, and its whole job is to shut down the activation of that pathway. But what you'll notice, they confirmed, they went and they looked at the human patients again, and those really big neurons have this phosphorylated S6. They have a really high level of phospho S6, and that's just a readout of how active this pathway is. So they're looking at it and they're like, cha-ching, this pathway is the problem in these patients. There's too much of it. So what are we going to do about that? We need a model in the laboratory to study this, to mimic it, so that we can look at new treatments. So this is where my project came in. First of all, why mice? There are actually a lot of laboratory animals that you could be looking at. We chose mice for a variety of reasons. One is that the genes are very, very similar between mice and humans. The other thing is that the development and organization of the brain are highly conserved. So if you were to look at these color-coded regions, in the human brain, they line up very well with the color-coded regions, functional regions in the mouse. So here was the goal. The goal was to take away the regulation of this pathway and then compare that to the disease, see what the mice look like. How on earth do you do that to mice? This is where genetic engineering comes into play. And this is not something I did. This was actually done by a collaborating lab. But I'm going to go through the quick process with you as to how this works. Essentially, you start with stem cells. You put those stem cells back into an embryo, and then you grow them up. So you make this artificial DNA. DNA is pretty cool. If you have the same sequence on either end, it likes to trade out what's in the middle. So by doing this, we can trade out the normal expression of this gene, that regulator, that P10 gene. We can trade it out so that it's no longer there anymore. But instead, what you get is this antibiotic resistance and a code for black coat. And that's important because you're growing up these ES cells, these embryonic stem cells, in a dish. And when you add antibiotics, the only ones that survive are the ones that you gave that new artificial DNA to. So now they're missing that functional sequence, but they have an antibiotic resistance. They also have this black coat, so that when you put them back in the embryo and put the embryos in a mother, she's a white mouse. She's going to have some white mice that have black stripes. They're going to have black portions. Wherever you see that black, you know that that mouse has some of that new DNA you put in there, some of that artificial DNA. If you make them some more, you're going to get some mice that are all black, and that means every cell and every tissue of their body has your new DNA in it. And that new DNA is expressing a black coat, and it doesn't have that P10 gene anymore. It's not functional anymore. So we've essentially removed that gene from their system. Pretty cool, right? This is actually really common. We do this stuff all the time. All right, this is why I like this. Okay. So these mice are different. You've genetically engineered them. Now, we have mice that are lacking P10. Do they look like the humans? Do they look like the patients? First thing we had to do is confirm that we really knocked it out. We call these knockout mice. And so if you look at human tissue again, this is mu N in green. It's a stain for neurons. Red here is that phospho-S6, that readout of how active the pathway is. And you can see where they overlap, it's nice and yellow. Those big neurons are expressing a lot of phospho-S6, right? We knew that already. The mouse does too. So this time in green, it's P10, and you'll see that a lot of these neurons still express it, but some of them don't. And the ones that don't are bright red with phospho-S6. So where that P10 is gone, you get a lot of activation of this pathway. Good, right? Mouse works. Do they have seizures? You betcha. All right? So just like you can put electrodes on a human's head and measure brainwave activity, you can do the same with mice. The only difference is that we actually implant the electrodes underneath the skull and secure it with this pink dental cement and actually plug them in because they like to scratch them off. They don't stay on. Mice don't understand to stay put when you're gluing things to their heads. Right? But when you do this and you watch for several weeks at a time, you'll see that they get more and more seizure activity. That's what these high spikes look like. So they have a progressive epilepsy. They look very much like the human patient. So now we have a good model to test new therapies, right? What do we want to do? We want to shut down the activation of this pathway. And luckily, there's actually a drug already available that does exactly that. And it's been approved by the FDA for other things. It's actually an immunosuppressant because it's a growth suppressant. So what we do is we implant them with electrodes to take a look at their seizure activity. We treated them for 2 weeks with either placebo, that's what vehicle is, or the drug, which is called rapamycin, and then we watched for several weeks to see what happened to their seizures. What do you think happened? Would I tell you if it didn't work? Come on. Okay, it worked really well. It actually worked really, really well. First, we looked again at that phospho-S6 to see what the readout looked like. You we saw that that green phospho-S6 was decreased, and also that really big cell size also went down. That also shrunk. That's what this shows you here. And the seizures almost completely went away. Almost completely. It worked really, really well. So when they were 6 weeks old, this was right after we finished treating them with the drug, their seizures were reduced almost down to nothing. 3 weeks later, we didn't give them any more drugs. 3 weeks later, it was still reduced almost down to nothing. And this got us really excited. Like, maybe we don't have to treat them forever, because this is a growth inhibitor, and we're talking about maybe treating children in the future. You can't stop kids from growing. Nobody's going to let you do that. It's not a good thing. So we got really excited about this, and we're like, okay, let's look even further. So this is just that same data plotted for you again. These are the mice that didn't get treatment. These are the mice that did. How many seizures they were having. It lasted a good long time. We started to get really excited until this happened. So then they started to get their seizures back. And we're like, well, so it's not a cure, but what if we treat them just periodically? We had a really good effect. Okay? So as long as we can continue to watch their seizures, if we could treat them every 4 weeks for 2 weeks, those seizures stayed suppressed and the epilepsy didn't come back. And this was really exciting, and it was my work and also work from other labs that did very similar experiments and similar models that led to clinical trials. This was just published last year. I wasn't part of this study, but colleagues of mine were. And what you'll see, if you just look at this one over here, is that of the 20 patients that were enrolled in this study, they all got the medication, and the majority of them had their seizures reduced by at least half, which is pretty fantastic. Remember, these people didn't respond to drugs at all before. So I hope what I've been able to show you so far is that by using these genetic techniques in these laboratory animals, you can mimic the human disease. They can benefit from treatments that are now targeted, and you can move it into patients as well. You can test it in patients, because you have to really test things in the laboratory first. You can't just go giving people drugs, unfortunately. It might be easier, but people don't like it very much. Okay. So this is my thesis research. It gave me a PhD. I was like, woohoo, I got a PhD. Now what? Right? Well, I told you guys before that I was interested in becoming a professor. And this is the traditional route that you would take. You would get your bachelor's degree, you go get your PhD, and then you continue doing some teaching and research at a university. And that's the route I took. That's where I am now as a postdoc. I'm working in someone else's lab, teaching and doing research. But there's a lot of other things you can do with a PhD in a biomedical science. One of the other more traditional routes is to go work in a pharmaceutical company where you can develop these drugs. But then there's a lot of other places you can go work too. You can work in policy, so down in DC you can work in the government, and you kind of get to decide what science is getting funded and what science isn't, and how much funding you're going to allocate to certain things like NASA or the NIH, that sort of thing. You can become a science librarian. This is actually really hot right now. So go be a science librarian. It might be a little lucrative. Okay, so there's lots of other things that you can do, but as I already mentioned, I wanted to continue my research. And so I wanted to take a different approach. I wanted to try this in vitro approach, where I could ask a little more detailed questions as opposed to the big picture questions. And so I moved from Texas to Philadelphia, and I joined another collaborative lab between Dr. Golden and Dr. Marsh. Dr. Marsh actually sees patients in the clinic at CHOP. He's a neurologist, he's a pediatric neurologist, in addition to having his laboratory. I did a bunch of research with Dr. Golden that I'm not going to talk about, but was also pretty interesting. Just in the interest of time, I'm going to tell you what I'm working on now with Dr. Marsh. So as I mentioned, he sees patients in the clinic, and he had a particular patient come in, a young boy who was 18 months old when he first came in, and he's just been devastated by epilepsy, by seizures, almost since birth. He's not responding to medication at all, he doesn't respond to light, he doesn't respond to sound, he doesn't respond to touch. And his parents were really at their wits' end. Like, can you imagine your toddler that can't do anything but have seizures all day. And he just, he requires constant medical assistance to survive. And so the parents decided to go ahead and do some DNA sequencing and see if they could find what the problem was. This is another one of those genetic disorders. And what they found was a mutation, a mistake, in the thorase gene. Everybody's calling out, what the thorase gene is. I don't know what that does. There is one group that has recently published a scientific paper about what this gene does. And so, remember I mentioned those neurotransmitter receptors and how they can be involved in turning up the volume, the excitation, or turning down the volume, the inhibition. So these AMPA receptors are involved in turning up the volume, okay? And when you find them on the surface of a neuron, that means that they can bind more neurotransmitter and turn up the volume even more. What happens when you're missing this gene, I don't know how well you can see that, is you get a lot more of these receptors sitting on the surface. Okay, so the gene must be involved in keeping things balanced. Without it, you get too many receptors on the surface. This is very interesting because we know something in neuroscience about how important these receptors are. When you're learning something, you get more of these receptors put on the top. This way they put on the surface. This way they can bind more neurotransmitter and strengthen that signal, that message. They can turn up the volume for that message. But if you're not really interested in what's going on and you tend to forget it, or your brain decides that's not really important information, I don't need to hang on to it, these receptors can come back down off the surface. Turns out that this gene that's mutated in this patient is really important for pulling them back down off the surface. Really important for turning down that volume. And this is what a representation of the response would look like if you could stick an electrode right in that neuron and measure what's going on. Okay? So this would be a normal response. This would be a really high response, turning up the volume. And this would be normal again. Well, when it's not there, you already know that you get too many of these receptors sitting on the surface, and so you get this really high volume. It's turned up too high. So this got Dr. Marsh thinking. He was like, well, if this is what's going on, because this is the mutation that the patient has, maybe we can do something about it. Maybe we can turn down that volume a different way. What if we give an antagonist? So an antagonist is a molecule that's going to bind very specifically to these receptors and not let go. So if it's bound to these receptors, now there's only one available for the neurotransmitter to hit on. And that should make it look normal again, even though there's extra ones sitting on the surface. Make sense? Okay. So we tried it. He got the parents' permission and he said, this is an experimental drug, hasn't been used like this before. I think will help your son. Guess what? The seizures are reduced. He responds to touch. He responds to light. He's still pretty devastated. He's not going to be a normal kid. But he's really improving with this medication. Which is fantastic, but the story's not over. Because we're not entirely sure what this mutation is doing, and we're not entirely sure how the drug is working. We have a good idea. This is the theory that I just showed you. That's what we think is happening, but we don't know for sure, and that's where I come in. So I'm going to test this in the lab in an in vitro way. So what I'm going to do is I'm going to take neurons from mice and culture them in a dish. And then there's some really cool genetic stuff I can do where I can take out their normal thorase gene and put his mutation in. And I can see what that mutation does. I can look to see how many of those receptors get put up on the surface. I can stick electrodes in those neurons and see what their responses look like, their electrical activity, how high or low that volume is. And I can treat with the drug that he treated the patient with. And I can see what the perfect dose is, and I can see if it's doing what we think it's doing. Because we want to be able to use this drug again, this medication, for patients in the future that might have this same mutation. We want to make sure it's doing what we think it is, and that it's not actually doing any harm in the culture dish. And so these are the kinds of things that we can look at. So that's what I'm working on now. I don't have any results yet. I can't show them to you, but that's the process that I'm going through in order to figure it out. Thank you. I was going to close a little. Okay. I just wanted to let you, let's skip to this one. That I hope I've been able to show you through these two projects and a little bit through my career path that, you know, what these neurodevelopmental disease models, what we're using in the laboratory, are really good for identifying and confirming the source of disease, which is what I did in these two cases, so that we can target new effective therapies. And now you can clap as I thank everybody.
So we have 10 minutes for questions, so take advantage of the time, and Nicole can answer any questions you might have.
Yes.
I'm sorry, I have laryngitis. How do people who are doing this kind of research communicate with each other when they have discoveries? When maybe one discovery in one area might be helpful. And the reason I ask this is because I want to get involved in nutrition research for PTSD. And a lot of these things that I hear, everything that I hear, it always goes right back to that place in my brain that goes, is that relevant? Is that going to help the PTSD? Is that something I might need to know for the future? And my question right now in my career path is, how do I meet people that are doing this stuff? How do I find out? How do scientists communicate with each other?
That is a fantastic question. So the short version of that question is, how do scientists get this information to each other? How do they communicate it so that you're not duplicating things, and so you're getting as much information as you can to make your next step, right? To be able to help your patients. There's a couple of different ways. One of them is conferences. So every year I try to attend a conference for the Society of Neuroscience, where 30,000 neuro nerds go and meet every year. There's only a couple places in the US that actually can house, you know, a couple convention centers that can house that many. It's usually 25 to 30,000 people that attend that meeting. And there's posters and there's presentations. And there's a lot of networking where people get to be friends with each other and they share information and they collaborate. But the number one way that this information gets transmitted is through publication. So every time you complete an experiment, you try to publish it in a scientific journal. So all the things I've showed you, except for the last story so far, has been published in a scientific journal. And that's how most of these journals are now. You can access them for free. There are some that you still have to pay, but a lot of these articles you can access for free on the internet. And so you can go through and you can find what's new in your area, and you search for certain keywords, like you would Google anything else, and you can find this information. And you can contact the scientists, the authors of the papers too. Lots of time they would love to tell you exactly what they've been up to and the cool new thing that they're working on now.
Any other questions? Please take advantage.
Did you find any side effects with the two drugs that you used with the first patient and the second patient?
Right, so the second patient, to my knowledge, there haven't been any real side effects. He's so devastated that any side effects would be so minimal at this point anyway. In the first case, so I mentioned that the drug, the rapamycin, is a growth inhibitor. And so we actually, when we looked at the mice, we looked at how they gained weight over time. And as long as we were treating them intermittently, they gained weight at a pretty steady pace. They actually gained weight better than those that weren't getting treatment because they were so sick they didn't eat. We think because they actually tended to lose weight. So there are some side effects, but they are minimal as compared to the effect you get on the seizures. Yes, in the back.
I guess I'm wondering why you're not going on to get an MD. PhD.
Oh, that's a good question. So originally I thought I might want to be a veterinarian. And I spent a summer working, actually I spent a couple summers working in a vet clinic. And as much as I loved seeing the animals every day, I kind of got bored giving vaccines and doing spay and neuter surgeries. And so that wasn't quite the career path for me. I thought about medicine. I thought about doing an MD because I really do like the patient connection. But patients, I consider myself a people person, but I'm not sure I could handle that many sick people. I think it would really be trying on me, especially since I'd be more interested in working with the pediatrics. These patients usually are not in a very good condition. And I I don't think I could actually handle that.
I saw patent law as one of like the job
Yeah.
details. Is that involved with the fact that like if this research is done, they prevent other researchers to do it the same way kind of thing? Or is
Not exactly. It's more like, so this particular drug, the rapamycin that I used in the first case, if we wanted to patent its use for epilepsy specifically, we could do that. But we need someone to help us do that. And so you can work in patent law and be kind of a science guy in the lawyer's office who looks at things and figures out if it's worth patenting, how to go about doing it, what kinds of wording you really have to put in there to make sure it works. So it's not, you're not trying to prevent other people from doing it. In fact, every time you publish your information, and you're supposed to publish it, any chemical you used, any organism you use, is supposed to be made freely available to the public. So you can patent things, you can work in patent law, but the idea is not to prevent other people from duplicating your work. Does that make sense? Yes.
So for me, the PhD sounds very lengthy in process and intimidating, and I wonder what other kind of people with smaller degrees, your degrees maybe, work with me. What's the support staff?
Oh, there's a lot of support staff. So if you have a bachelor's degree in biology, you can work in a laboratory as a technician. And actually, the technicians run the laboratories. Let me be completely honest with you. The rest of us don't know how anything works. The technicians keep things going. They are really good at the lab bench. They're really usually they're very technically savvy. So you can do that. You can work in the lab and be part of these projects. I have a colleague who has a master's in biology, and she makes more money than I do. She worked in a laboratory for a while. She became what is known as a lab manager. So she literally ran the lab for everybody. She was your go-to person if you needed to buy anything. She was your go-to person if a piece of equipment wasn't working. And she now works in administration and helps people write grants for money so they can do their research. So there's lots of area in between.
On many.
Yes.
Not anything.
Very much, yeah. Yeah, there's a lot of other avenues if you're interested in biology and chemistry. You usually have to start with your bachelor's degree. There's lots of options, even with the bachelor's degree, without going further.
Question.
Yes.
My long-term career aspiration is to be a classical reconstructive surgeon, but I've always been interested in like neurology and neuroscience and I've wanted my PhD. So I'm wondering if you think it's possible to do both with the amount of time it takes to reach them.
You can. So I know people who have gotten combined degrees, MD, PhD, and it is a long process, but where those combined degrees are available, they usually try to cut it down for you. So it took me 6 years to do my PhD. The average is about 5. To do your medical degree is usually about 4. If you combine the two, you can do it in about 6. Okay, so that's not too bad. But when it comes to medicine, you also have your fellowships and internships and residencies, whatnot, afterwards. But you get paid a little bit better to do the work then. So you're looking at least a 6-year in school, and then your fellowships after that.
And just to add to that, my doctorate took 9 years because
Yes.
I was married and working full-time at the
Yes.
same time.
Yeah, they can be very variable. I know some people who did theirs in 3, so it has a lot to do with your personal situation.
Yeah.
Yeah.
What would you say is your most optimistic view of neuroscience at this point? Like 50 years from now, are we looking at super brains? Where's the golden, like what could all this lead to in the long term? I know you guys are really on the uphill battle right now, but where's the horizon?
Well, you'll hear people say that, the brain is like the last frontier. This is this is where all the discovery is going on. And I don't know if you guys are familiar, but Obama actually put out this brain initiative last year where he wants to map all of the connections in the brain. And so there's a lot of funding right now in order to do these things. Are we going to be able to put our brains in computers? Maybe. I mean, I think a lot of the stuff that people talk about as being possible is possible. Maybe not on the timeline you're thinking about, because the 86 billion with 1,000 connections each, that's complex. It's a little hard to figure out. but we are making dents in it, and there's a lot of cool things we can do.
Well, we're at noon now, the end of the talk. You might want to stay and ask her some personal questions, but if you didn't sign the roster, I'll put it up here. I'd like you to sign, and let's thank Dr. Soon.